Pathogens. Infectious agents that cause disease. Lysozyme. Enzyme that digests microbial cell walls. Phagocytosis. Ingestion of bacteria and other foreign substances for cellular defense. Antimicrobial peptides. Substances that disrupt or kill fungi and bacteria by disrupting their plasma membranes. Toll. Protein receptor on the surface of immune cells that leads to the activation of the nucleus to produce a particular set of antimicrobial peptides. TLR. Receptor that recognizes fragments of molecules characteristic of a set of pathogens. Neutrophils. Most abundant phagocytic cells, these engulf and destroy microbes. Macrophages. Large phagocytic cells that sometimes reside permanently in various organs and tissues to combat pathogens. Eosinophils. Phagocytic cells low phagocytic activity but poison outside invaders, especially parasites, by positioning themselves against the parasite's body and then discharging enzymes that damages the invader. Dendritic cells. Phagocytic cell that stimulates development of acquired immunity. Interferon. Protein that provides innate defense against viral infections, limits cell-to-cell spread of viruses in the body. Complement system. Consists of 30 proteins in blood plasma that function together to fight infections, activated by substances on surface of microbes. Inflammatory response. Changes brought about by chemical signals released upon injury or infection. Histamine. Important inflammatory signal that triggers blood vessels to dilate and become more permeable. Mast cells. Connective tissue cells that store histamine. Pyrogens. Sometimes I cry. NK cells. Recognizes and eliminates certain diseased cells in vertebrates, causes cell death in cells that are diseased in the body. Lymphocytes. Another name for white blood cells. Thymus. Where T cells mature. Bone marrow. Where B cells mature. Immunological memory. Enhanced response to foreign molecules encountered previously. Cytokinase. Proteins that help recruit and activate lymphocytes. Antigen. Any foreign molecule that is specifically recognized by lymphocytes and elicits a response from them. Antibody. Secreted protein from plasma cells, soluble form of antigen receptor. Ig. Another name for an antibody. Plasma cells. B cells can become these types of cells that produce antibodies. Epitope. Small, accessible portion of an antigen that receptors and antibodies just recognize. B-cell receptor. Receptor that is Y-shaped, consists for 4 polypeptide chains, 2 heavy chains and 2 light chains. T-cell receptor. Receptor that has 2 different polypeptide chains, alpha and beta chain. Class I MHC molecules. MHC molecule that is found on almost all cells of the body. Cytotoxic T cells recognize displayed antigen and then destroys the sick body cell that presented the antigen. Class II MHC molecules. MHC molecule that is only made by a few types of cells, displays antigens for recognition by cytotoxic T cells and helper T cells, does not destroy body cell that presented the antigen. Clonal selection. When the presentation of an antigen to specific receptors on a lymphocyte leads to repeated rounds of cell division. Humoral immune response. Response that involves the activation and clonal selection of effector B cells that secrete antibodies. Cell-mediated immune response. Response that involves the activation and clonal selection of cytotoxic T cells. Helper T cells. Cells whose role is to secrete cytokinase to activate B and cytotoxic T cells. Neutralization. Process where antibodies bind to surface proteins of a virus or bacterium, blocking the pathogen's ability to infect a host cell. Opsonization. Process where antibodies bound to antigens promotes phagocytosis.